I would like to keep this educational, so I'm going to talk about iPS cells, and please ask whatever details you want at the end of the two talks or as you see fit. So reprogramming, the first thing that has taught us is that the potency that is lost during development, as you see the first cell, the zygote that starts development, that's a pluripotent cell. It will give rise to all the cells in the embryo and to part of the placenta. But that property, this potency, developmental potency, is lost throughout development. And we thought for many, many years that that was something irreversible. We could not regain this potency. And that's, in fact, one of the basic principles of developmental biology that is represented in this way, that developmental potency is lost gradually upon development. What we have learned is also that this potency can be regained, and one of the experimental paradigms that have taught us that is induced reprogramming. And when we do induced reprogramming to pluripotency, we take somatic cells, cells that have a very established identity and no potency whatsoever, and we bring them back in their developmental history to cells that have a potency equivalent to a cell in the inner cell mass of the blastocyst. I mean, a cell that is pluripotent, it still has the capability to give rise to all the cell types in the embryo. Plus, we give them self-renewal. These cells, the cells in the early embryo that have no self-renewal ability, of course. In vitro, we can give them self-renewal ability, so we obtain cells that are pluripotent and are stem cells. We can maintain them in vitro for indefinite periods of time. So this is what we learned from the seminal work of Shinya Yamanaka's lab in 2006 in the mouse, where he took mouse embryonic fibroblasts, in this case, cells that were specialized, and by doing the tricks that he did, which he called induced reprogramming, he reprogrammed those cells to look like this cell type here, which this is a colony of something that looks very much like mouse embryonic ES cells, and behave in the same way. They are able to differentiate into cell derivatives of the three main germ embryo layers, definitive endoderm, ectoderm, and mesoderm, so they are pluripotent, plus they are able to contribute to the germline and to generate, you can take one of these cells, which he called induced pluripotent stem cells, and generate an entire mouse, proving that they are indeed pluripotent. Very shortly afterwards, this is the end of 2007, beginning of 2008, three different groups, independently, including that of Yamanaka's, show that the same procedure also works with human cells. So you can now take somatic cells, human somatic cells from any person, and reprogram them to behave as human embryonic stem cells. Just keep one thing in mind, that is, this is how embryonic stem cells look like in humans. But keep in mind that we do not have a very stringent test to assess the pluripotency of these cells in contrast with the mouse. So the best way we have to assess the pluripotency of human iPS cells, or human ES cells for that matter, is something that we call a teratoma assay, which is basically inducing a tumor. I will talk about this a little bit later. But just wanted to keep you in mind during the rest of the talk and during whenever you hear about human iPS cells, is that it's very different to compare a tumor to assess a tumor than to assess a living fertile mouse. So we really don't have a way to tell that our human iPS cells or ES cells are truly pluripotent and are truly reprogrammed. So keep just that in mind for the future. The way that we assess these cells in the lab, the way that we characterize, is these cells are stem cells and are pluripotent. So first we test that they are stem cells. Stem cells is just a cell that is able to divide indefinitely. Of course, indefinite is not a very defined term. When we use stem cells in animals, we typically refer to the lifetime of the animal. This is the indefinite. In the lab, any cell that you put in culture undergoes cell senescence, and you can observe that between 15 and 20 passages in vitro. If your cells are able to propagate, to be passaged beyond those 15, 20 passages, that means that your cell has escaped senescence and is able to self-renew. And if it maintains a degree of potency, then it's a stem cell. So, as I said, how we check that they are self-renewing in the lab? Very easy, just propagate them. If you can propagate them more than 15, 20 times, then your cell has self-renewing ability. And stem cells do that maintaining their karyotypic stability as opposed to cancer cell lines. To check that the potency that these cells have is pluripotency, we check a series of markers that are very typical. I'll show some of them. We also check in vitro in the lab that these cells are able to differentiate, and they can differentiate into derivatives of endoderm, ectoderm, and mesoderm. And we do this test that I talked about, this teratoma assay that I'll show you how it looks like. This is basically the same characterization that we do for any embryonic stem cell. So the cells that we derive from pre-implantation embryos, we subject them to the same type of tests. For iPS cells, in addition to this, we typically fingerprint the DNA of these cells, so we double check that they come from the original, the patient, or the original source of somatic cells. We check that they are genetically identical. If we use reprogramming transgenes that integrate in the genome, we may want to check that they have integrated and where they are, and they don't affect any endogenous loci. We don't typically use integrating transgenes to reprogram anyway, but you may want to check that they are there if you were integrating them. But in any case, whatever method you use to reprogram, you have to make sure that if it is an integrating method, your transgenes are no longer active, they have been silenced. And if you use a method that doesn't integrate and that the transgenes or the vectors you use are cleared from the cell, you need to make sure that those are out of the cell. The reason for this is, or for the importance to check this, I think is double. First, iPS cells or stem cells in general, embryonic stem cells, they are very, very good at silencing any proviral promoter, okay? Something that somatic cells typically don't do. So when you are reprogramming using viruses, when you are reprogramming a somatic cell, say a fibroblast, your transgenes are expressed, driven by the viral promoter. But as soon as this cell becomes pluripotent, first thing it does is silencing those promoters, silencing those transgenes. If you check your cells and they have not silenced the transgenes, that means that they were not fully reprogrammed. And second, if you have a residual expression of the reprogramming transgenes, some of them I'll show are pluripotency inducing factors. So your cells will have problems differentiating. Okay? They will be maintained in an undifferentiated state artificially. Okay, so that's important. I'll also show how we do that. And in the end, we want to check that these cells that we have reprogrammed are reprogrammed in terms of transcriptional landscape. The genes that are expressed and the genes that are not expressed in those cells are totally different from the cell we started with, and we need to check that. And also the epigenetic landscape of the cells that we have reprogrammed changes. Okay? So how all of this looks like in the lab is, this is how these cells grow. In the human, they form these large flat colonies, which we now know as primed ES cells, but they look like this. They typically grow on feeders, although there are various methods to grow them. They have strong alkaline phosphatase activity, like many other cells do. And they express a series of markers that are very typical. None of these markers is exclusive of embryonic stem cells or iPS cells, but in these combinations they're very, very characteristic. And there are some specific surface markers such as TRA181, TRA160, and also some of the SSCA family like SSCA4 or SSCA3, and also transcription factors such as SOX2, NANOG, OCT4, etc. Now you can get this as a kit. I mean, there are several companies that sell you kits with the antibodies to characterize this type of cells. These are iPS cells are karyotypic stable, so after several passages, more than 15, 20 passages, typically you check their karyotype and check that they are, you can check by normal G-bending karyotype or use other more sophisticated techniques, but check that they can be and should be maintained with a normal karyotype. Sometimes they acquire, like any cell that you put in culture, they may acquire abnormalities, but it's not typical of these cells. This is something that you did, and you should go back and start with a fresh batch of cells that didn't have any abnormalities. So they can be maintained free from any karyotypic abnormality for hundreds of passages. And it's good to check it from time to time. This is, the graph is just showing the expression of the transients that we use to reprogram. Those that say trans here, like the transients that we use for these cells were OCT4, SOX2, KLF4, and MYC, which is a typical Yamanaka cocktail. You see that the expression of the transients has been silenced, and the expression of OCT4 and SOX2 now comes from the endogenous OCT4 and SOX2 loci in the cell. And several other genes become expressed, such as crypto, REX1, NANOG, which are genes that are typically expressed in pluripotent cells, showing that these cells that you have, these iPS cells, are expressing the genes that a pluripotent stem cell is expected to express. These are just a handful of genes, but you can also check genome-wide, and you see that the expression of all the genes in iPS cells is very, very similar to that of ES cells. This is just a comparison of genome-wide expression profile in iPS versus ES cells. There's some outliers, as you can see, but if you check different clones, you find different outliers. In general, the expression profile of iPS cells and ES cells is very much indistinguishable. And in terms of epigenetic reprogramming, the easiest thing to do, and what we routinely do in the lab, is check at methylation, promoter methylation. Genes that are not expressed, they are typically repressed by strong silencing induced by methylation of the DNA in the regulatory regions. So, for instance, OCT4, if you're not familiar with this type of representation, each circle is a CpG, and if it is empty, means that it's not methylated. If it is filled, means that it's methylated. Okay? So you see that two genes, the regulatory regions of two genes such as OCT4 and NANOG, these are genes that are typically expressed in pluripotent cells, but they are not expressed in somatic cells. So in somatic cells, like fibroblasts or keratinocytes, they are not expressed, and the regulatory regions are heavily methylated, keeping the silencing of these genes. If you take these same cells and you reprogram them, those regulatory regions become demethylated. So the expression of these genes is maintained in these cells and is no longer repressed. This happens at the OCT4, NANOG loci, and at any other loci. The entire epigenetic landscape of the cell changes to a cell that is of pluripotent identity. Okay. For differentiation, the easiest way to differentiate these cells, there are many directed differentiation protocols, but just to characterize the cells, what we do is form these aggregates here, what we call embryoid bodies. And these are really like, mimicking what happens during early embryo development. They are just aggregates of cells, but when you do that, cells start doing something similar to gastrulation, and they form mesoderm, and you can find derivatives of endoderm, mesoderm, and ectoderm in those bodies. So if you let them differentiate for long periods of time, you can find structures that look like glands and stain for markers specific for derivatives of definitive endoderm in this case, so you can be sure that your cells have the ability to form endoderm, same for mesoderm or ectoderm. So if you can do that with your cells and cells that you have derived in a clonal way and maintained like that, then you are sure that your cells have at least the ability to differentiate into these 3 embryo germ layers, and then we can conclude that they are pluripotent. So the thing is that in vitro you can do many things, and you can force cells to differentiate to fates that they are not inherently prepared to. I mean, just tricking the cells to differentiate may not represent their potency. This is why we do these teratoma assays, although, to be honest, we don't do that anymore, but it's in the books, and I think we should tell you. But in this case, what we want to know is what is the endogenous ability of these cells to differentiate. Okay? So what we do is put them in an environment that will not be instructive in terms of developmental signals. And for that, we use a mouse. We use a mouse that needs to be immune deficient, so they don't reject the cells, which are of human origin. And we put them under the skin or under the kidney capsule in an environment of an adult mouse, an environment that will not provide developmental information. In that environment, the cells get nutrients, they get vascularized by the host, and they start proliferating and form a tumor. But this tumor is like an embryo completely in disarray. So you will find bits and pieces of structures. So you have an eye that is not organized. You have a piece of neuroretina in one side of this tumor. You have a piece of cartilage, a piece of muscle. And if you have bits and pieces of derivatives of the 3 embryo germ layers, and you don't have any obvious bias, like most of the structures that you see are from mesoderm, but you see that roughly 30% of all the structures are from each embryo germ layer, then you can conclude that your cells have the ability, the endogenous ability, to differentiate to any lineage with no particular bias, and conclude that your cells are pluripotent. So this is how the tumors look like. They can be quite big. They're benign tumors. The mouse is just uncomfortable because of the size of the tumor. And these are all sections from the same tumor where you can see structures by H&E staining, or you can do immunofluorescence and find the same. You see neural rosettes with markers of neuroectoderm, or you can find the same glands with that stain for markers specific for endoderm or pieces of muscle. Okay, so again, if you find all of this, you can conclude that your cells are pluripotent. As I said, after many years of doing teratomas, we haven't really found any case where the teratoma told us that the cells were not pluripotent, that we hadn't catch in earlier tests. So if the cells meet all the criteria that we have been testing, say, including in vitro differentiation ability, they meet the in vivo differentiation ability test always in our case. So, and you use an animal to do this. So in the end, at least in our center and many other labs around the world, have decided that we will not use mice anymore to do this test. Okay? Anyway, the way we do it in the lab right now, reprogramming itself is quite easy. You just have to introduce these 3-4 reprogramming factors. As I said, the original Yamanaka cocktail is formed by KLF4, SOX2, CMYC, and OCT4. CMYC is dispensable, so you can use just the 3 other factors, introduce them into the cell. Yamanaka originally used gamma retroviruses, but many other viruses, integrative and non-integrative, have been used, plasmids, mRNA, even protein. Introduce them into the cell and maintain them in the cell active for a short period of time. I'm talking about days, less than a week, into your somatic cell of choice, for instance, dermal fibroblasts in this example, and after month and a half in the case of human cells, the cell you started with is completely reprogrammed and start behaving like a human embryonic stem cell. It looks like this type of colonies that we then characterize as iPS cells, human iPS cells. So this technique is very, very robust. Hundreds of laboratories around the world have used them, used the technique with success, has been used to reprogram fibroblasts, keratinocytes, and any cell type that you may think, and it works. It has been used to reprogram mouse cells, human cells, and cells from many different species, showing that reprogramming is a technique that is of general use and I think highlights a general principle of biology that you can change the identity of a cell using a set of transcription factors that are instructive for that fate. And I'm sure we'll hear more about that later. What I wanted, this being an educational session, to tell you that it's really reproducible, it's really robust. You can buy this technology now in a kit for reprogramming with your method of choice. What are the different many labs that are using this technology, using it for? Say there are 3 main applications of this technology, and I will today talk only about one. The first is just to understand the process of reprogramming. Basic research. Reprogramming is a very interesting process in itself, and studying induced reprogramming is giving us a lot of information about how pluripotency is acquired, how pluripotency is maintained, but also how cells can escape senescence and become self-renewing. And there are many labs using induced reprogramming as a tool to understand this type of questions. More biomedically applied is, since we can generate iPS cells from any person, including diseased people, including patients, there are some diseases that take place because a specific cell type or organ fails, and these are what we call degenerative diseases. In those cells, and which are the diseases that are most prevalent in the world, by the way. So in those diseases, if we could make that cell type that is failing or is diseased and put it back in the patient, we could cure at least the symptoms of the disease. So this is the rationale behind regenerative medicine, the concept of regenerative medicine, and iPS cells will be a perfect tool because we can generate them from any patient, and then we can make cells that are exactly genetically identical to those of the patient to differentiate them into the cells that are missing in the patient and replace the cells that have been lost in the patient. In addition, with these cells, since you remember I said that you can maintain them in the lab for very, very long periods of time, so they're amenable to manipulation. We can play with them and we can change quite easily their genome. So if the disease that is present in the patient is caused by a genetic mutation, we can play in the lab with those cells and correct that mutation. So the cells that we generate are genetically identical to the patient, except that they do not contain the mutation, so they will not suffer the disease. So this is something that, if you have been following, the field has been advancing very rapidly. There's a clinical trial, phase 1, just toxicity testing ongoing right now using this type of strategy, and we'll probably hear more in the future. There are, of course, problems, but for this talk today, wanted to focus on some side effect of this technology that we have found. And is that most of us, I think, we started trying to make cells to cure patients. But what we saw is that when you took those cells from the patient, made iPS cells from the patient and then differentiated them into the cell that was relevant for that patient, we found that those cells were sick. So we could study how those cells got sick in the lab. And this is the first time that we could do that with human cells. So we started creating models. And I think this iPS cell technology has become a very, very useful way to create models that had not been previously available to study diseases. This is particularly relevant for diseases, or has been mostly exploited for diseases that are of clear genetic origin. I mean, monogenic diseases are quite easy to model using this technology. And I'll show you some examples. The principle behind this type of approach is that you take a somatic cell from any patient, a cell that will be easily accessible, typically a skin biopsy, but you can use many other cell types that are easily accessible from the patient, generate iPS cells, and if the disease is in any way genetically linked, if there is something in the genome of that patient that is predisposing the patient to suffer the disease, then that predisposition will be captured in the iPS cells because they are genetically identical to the patient. Okay? And then you can differentiate those cells, as I said, to any cell type in the body, so including the cell that will be relevant for the disease of the patient. And if you have that cell, you can analyze if the cell shows any phenotype that will be reminiscent of what happens in the patient. Phenotypes have been found in many, many diseases. I think this is one example that we did a long time ago using Fanconi anemia. Fanconi anemia, typical monogenic disease. If you have a mutation in one of the genes of the Fanconi anemia pathway, you have Fanconi anemia, and I don't know if you're familiar with the disease, but you have a problem in the DNA repair machinery. So one way to test that, and these were experiments done by Jordi Surrayes here in the Universitat Autònoma. They have this very clever way to check the pathway. This is the nucleus of a fibroblast, by the way. Okay? So what they do is they hit with a very high energy UV radiation, but that is very, very focused. So you can damage the DNA at a very specific position in the nucleus. So when you do that, if your DNA repair mechanism works, you have some proteins, for instance, FANG-D2, go into these sites and repair the DNA. If Fanconi anemia pathway doesn't work, then this mechanism doesn't work, and you see this is what happens in the fibroblast of a patient, that this FANG-D2 is not recruited to the sites of DNA damage, okay? Because showing that there is, this is a phenotype that you find in the cells from this patient, that their DNA repair mechanism doesn't work. So these are fibroblasts obtained from iPS of those patients that have been corrected for the disease. They've been gene complemented, and you see that upon hitting the DNA, you get the pathway FANG-D2 being recruited to the sites of DNA damage. So you can study the phenotype of the disease obtained in cells derived from iPS of these patients. So this is one of the first times that we saw that these cells that we obtained from iPS from patients could be sick in vitro, and we could even cure them and see if different treatments will cure these cells instead of using the patient to do that. Many other diseases have been modeled. This is Gaucher disease. In this case, critical thing is that you need to produce the cell that is relevant for the disease. In this case, one of the cell types that is affected is macrophages. So in macrophages of the patients, we see that there are very few when compared to control or to gene-corrected iPS cells, few macrophages expressing high levels of GBA, and that can be corrected. So there is a phenotype in these cells when you get the cells that you can also see in the patients. In collaboration with Danny Grimberg and Luisa Villageliu, and this was the work of Isaac Canals, a very talented PhD student in the lab that is now with Henrik. We modeled Sanfilippo. In this case, again, the trick was what is the cell that we should be looking at, the cell that gets sick in the patient, neurons. So if you take iPS cells, make iPS cells from these patients, differentiate them to neurons, you see these are neurons, electron microscopy of a neuron. From a normal person, you see the lysosomes, how they look like. This is how the lysosomes become in the neurons from these patients. These are neurons that we have generated in the lab, because we cannot typically obtain the neurons directly from the patients. And even if we could, which we do normally post-mortem, even if we do, we could not see the cells getting sick. You just get cells that are already sick. And in this case, for instance, we could see how these abnormal lysosomes become more and more cured with time when we complemented these cells with the gene that is missing in these patients. So you can test treatments and see if they work. Okay. We can also test things that you cannot test otherwise. This with hemophilia is a project we've been doing with Jordi Barquinero at Valdebron and Fran Vidal, and also with Anne Weber at INSERM. In hemophilia, you know that in this case it's hemophilia B, where factor IX is mutated, and this gene is only expressed in hepatocytes. There is no way to check the expression of this gene in any other cell type. So the mutation in this case in particular was a problem with that is supposed to affect an alternative splicing site. So the only way to check if this is true is to express the gene, and you can of course do that with many genes and heterologous expression systems, but you never know if 293 or COS7 are going to use the same splicing machinery that a hepatocyte is using. So here you get iPS cells from the patient's proper controls, generate hepatocytes, and study the expression of this gene. And we can see that this is the mother of the patient, where the vast majority, 99.9% of the transcripts, are using the right splicing site, whereas the patient, only has 2.9% of the transcripts using the right splicing site, whereas the vast majority of the transcripts are wrong and give rise to a protein that is truncated. Okay. But if you realize all the cases that I've mentioned where diseases that are monogenic, and there are, I showed 2 or 3, but there are hundreds of diseases that have been modeled using iPS cells so far. But most of them are monogenic diseases and diseases of very early onset or congenital. So if you're interested in a disease, in a monogenic disease of early onset, iPS or reprogramming in general is a very good way to test. Most likely you will have a cell which shows the phenotype that you see in the patients. Okay? But most prevalent diseases are not, of course, monogenic diseases of early onset, are quite the opposite, are diseases with a very complex genetic influence and diseases that appear late in life. So we decided to check, and other groups decided to check, if this reprogramming technology can be also used to model this type of diseases of late onset and complex genetic predisposition. And we chose Parkinson's disease for that because it's typical, it's a very frequent neurodegenerative disease. It takes decades to evolve in the patient, so it's a very progressive disease. And like any other highly prevalent disease, most of the cases are sporadic. We cannot find any genetic predisposition, any clear genetic predisposition in those patients. But there are, like any other highly prevalent disease, there are several cases where the disease runs in families. In the case of Parkinson's disease, is about 10–15%. And when you look at those families, you do find genes, the mutation of which is associated to increased risk of suffering the disease. And in the case of Parkinson's, there is a number of genes. Just want you to focus on this gene. LRRK2 is the most prevalent cause of familial Parkinson's disease. So we decided to see if we could model Parkinson's disease in the lab. And for that, we recruited controls, we recruited patients with sporadic Parkinson's, and we recruited patients with Parkinson's associated to mutations in that gene. This is a typical report that we generate from an iPS cell line, where you see, as I introduced earlier, they express pluripotency associated markers. They can differentiate in vitro, generate teratomas. Still, we did teratomas at the time. Karyotypically stable, genetically reprogrammed, transcriptionally reprogrammed. Oops. And in this case, this is a line from a patient with a mutation in the LRRK2 gene, and you can see that the mutation is in the iPS cells, of course. So overall, we generated, I think, about 55 lines covering these 15 individuals. And when we generated dopaminergic neurons, which is the relevant cell type for this disease, we saw that all of them generated dopaminergic neurons nicely and with no difference in numbers or in characteristics. We didn't expect any difference because, of course, these patients were born with their dopaminergic neurons in place and worked for many years. So we didn't find any difference. The only thing that we found is that, I don't know if you can see here, that alpha-synuclein is abnormally accumulated in dopaminergic neurons from the patients with mutations in LRRK2, not with sporadic patients. And we saw that for most of the dopaminergic neurons derived from iPS cells from these mutant patients had abnormal accumulations of alpha-synuclein. Again, not extraordinarily surprising because it had been shown that alpha-synuclein and LRRK2 have a direct genetic interaction that had been shown in mouse models. So the fact that mutant cells showed alpha-synuclein accumulation was not surprising. But having these cells, having the dopaminergic neurons, gave us, from these patients, gave us the opportunity to look at what is doing the mutation in this particular gene that makes alpha-synuclein accumulate in these cells. And in collaboration with Ana Cuervo at Albert Einstein, we found that LRRK2 is being normally degraded using a degradation pathway very peculiar known as chaperone-mediated autophagy. And this is a pathway, a degradation pathway, that is shared with many other proteins in the cell. When LRRK2 is mutated, it not only blocks the pathway, but it poisons it. So it doesn't degrade other substrates of the pathway, including alpha-synuclein, which is another substrate for this pathway. And this is why alpha-synuclein becomes accumulated in these cells. And this is something that you can do when you have the disease-relevant cell type, I mean the cell that is experiencing the disease with all the proteins that should be in that cell, coming from the patient with the specific peculiarities of the patient. So this, okay, these cells, okay, I'll be quick. It's very difficult to reproduce in the lab within the time frame of a normal experiment, something that takes many, many years to take place, to occur in the patient. But we can do it. If you age these cells, if you let them in culture for up to 75 or 90 days, up to 3 months, they start degenerating. Okay? But they degenerate only or more evidently the ones that come from the patients, not from the controls. Okay? So they have an increased susceptibility to undergo neurodegeneration if you culture them for a long time. And I'll skip this if you, because I think what I will remark from this technology as an advantage, before going to the disadvantages, is that over the years that we've been using this technology to model diseases, what we have learned is that what we generate here, these disease-relevant cell types that we differentiate from iPS cells, what truly represent are the cells that the patient had when he or she was born, okay, much earlier than the disease appeared. And when you use this to your advantage at modeling chronic progressive diseases, you can start looking for phenotypes that you never saw in the patient. Okay? Because when the patient arrives to the clinic, there's already some problems, but you can see these cells get really sick in the lab and check things that had never been seen in patients. This is something that I think was very, very nice that we did with Danny and Luisa, and also in collaboration with Jordi Soriano at the UBA. These are neural networks that we organize from neurons differentiated from iPS cells from patients. These networks, you see how cells communicate with one another, and I mean, if you see something, please tell me. I don't see anything. I just see, light going up in some of them, but you can see how some respond, some don't respond. If you give this to somebody, this is Jordi Soriano, he's a physicist specialized in information theory, that can take this information and obtain meaningful information on how the network is synchronized and how it operates. And he studies a particular property of the network, that is how firing, a neuron firing, is able to influence the probability of neighbor cells firing. Okay? This is what he calls effective connectivity. And when you look at this property in neural networks obtained from normal cells, from neurons obtained from healthy individuals, this is the behavior of the network, where you see the density of the lines indicates the activity of the effective connectivity of the network. This is the same information obtained from networks organized from patients, in this case of Sanfilippo, where you can see that there is a huge decline in activity, in the functionality of a network that otherwise looks normal. The cells, in this case, they look normal, they don't show any problem. And how you can correct this? Just by correcting the genetic defect, the network goes back to normal. That tells you that not only you can find phenotypes that you have seen in patients and study the molecular mechanisms that are behind those phenotypes, but you can also obtain phenotypes that have not seen in patients, phenotypes that predate the onset of the disease, and then screen not only for drugs that will revert the phenotypes, but even for drugs that will prevent the disease from appearing. Problems with us. I will conclude that with this very quickly. Not everything needs or should be modeled using iPS cells, of course. Big problem is the cost. It's very expensive to work with these cells, and in many cases, it's just not worth it. If you have a phenotype that you can score in fibroblasts from the patients, don't make iPS cells, just use the fibroblast. Then is don't make iPS cells of diseases that have already been reprogrammed. Just ask whoever made those iPS cells to share. And we're sharing, and most people are sharing with no problem iPS cells from different diseases. There is a problem in to what extent a cell line that you generate from a patient truly represents the patient. And this is at two levels. Whether it represents the patient, and now it comes the issues of mosaicism, somatic mosaicism in the patient. Only thing we can do is generate several clones from each patient. Okay? If you do that and you have a phenotype that is consistent across several clones, then you are certain that that phenotype is representing the patient. But not all the patients are representative of the disease, so you should need, standard in the field is 3 clones per patient, at least for any new disease that you model, at least 2 patients and 2 controls per disease. One way to make sure that if you're working
Sorry, Angel, you should be finishing, because if not, Henry wouldn't be able to talk.
I'll be happy to discuss then more problems. Thank the people, the funding, and you for your attention. Sorry for the... Please, your turn.